So we should really start by thanking the organisers for the amazing job that they've done to bring us all here to Barcelona. They can't thank themselves, so we should do it on their behalf. Okay, slides. Should I escape and put my own slides up? Yeah. Okay.
You can, you're good.
Okay.
You just make it, yes.
Okay, good stuff. So I'm going to talk about some of the work that we've been doing to really try and understand nucleotide variation in cancer genes and to do so at scale. So there's been around 30 years of cancer genetics so far, and yet the two questions still remain. Who is it within our population that is at greater risk of developing cancer? And when a cancer forms, what are the genetic variation that contributes to the development and the progression of that disease and also the resistance to therapy? So in the context of germline predisposition, we know that around 10% of people who present in a cancer clinic will have a germline predisposing allele. We know that in conditions like ovarian cancer, the frequency is around 20%, and in other malignancies like renal cancer, the frequency is around 5%. We also know what some of those genes are. For example, in ovarian cancer, we know that there are genes like BRCA2 and RAD51C, and in renal cancer, it's genes like BAP1 and VHL. And there are also other genes like P53 and, as I'll describe, POT1 that can also contribute to risk, and they can do so across a range of different cancer types. So why do we care about this? If you can identify those people within a population who are at greatest risk, you can place those people in screening programs, and we know that this improves patient outcomes. There's a wealth of evidence to suggest that this is the case. In addition to this, what we can also do when we identify those individuals is identify other members of their family who we can place in screening programs. They can undergo cascade testing, which can in turn allow those individuals to enter screening programs and potentially also to receive prenatal testing as well. So our introduction to cancer genetics really began here. This is a pedigree from Leeds in the north of England, and the individuals that you can see who are shown here in black, these are individuals who've had a diagnosis of melanoma. We should compare this to the population frequency of melanoma in the UK, which is around 1 in 50. So clearly, the individuals within this pedigree are at much greater risk of developing the disease than others within the population. So the sequencing work that we've been doing over many decades has told us that about 40% of these individuals will carry variants in a gene called CDKN2A. Some will carry variants in genes like BAP1 and CDK4, but around half of these patients cannot be explained by variants in these known genes. So what we've been doing is trying to identify these new genes. And about 10 years ago, we discovered a new player. This is a gene called POT1, also known as the protection of telomeres 1 gene. So this was the first pedigree that we identified from one of these families. And you can see all the individuals here in black are individuals who've had a diagnosis of melanoma. And just to give you a sense for the penetrance of the disease, this individual here, by the age of 30, had 8 independent melanomas and actually had died of the disease. Here's another pedigree here. There are 4 melanomas in this individual by the age of 50. So these mutations powerfully predispose an individual to develop cancer. So in addition to the germline variants within this gene, the other defining molecular feature of POT1 is that these patients have enormously long telomeres. So this is a graph here that shows telomere length. The people shown here in grey are controls, and the ones in teal are the ones that carry those germline variants within POT1. So the telomeres in these individuals can be 3 or 4 times longer than others within the general population. So over the last few years, we've learned that individuals who carry these germline variants are about 7 times more likely to develop melanoma than the general population, and it's also been linked to a range of other malignancies. POT1 has also been defined as the first of what's called a long telomere syndrome by the World Health Organization, and if you present in a melanoma clinic anywhere in the world, including here in Barcelona, with a family history of melanoma, you will be tested for a variant within POT1. So as I've said already, POT1 is the protection of telomeres 1 gene. It regulates telomere length, And it also has a second function, which is involved in protecting telomere ends. And it's part of the complex you can see shown here, which is called the Sheltram complex. And the way to think about this complex is that it protects the ends of your telomeres from becoming frayed and damaged. And when the telomere ends become frayed and damaged, this can cause chromosomal rearrangements, which can in turn promote tumorigenesis. And in this context, all of these different types of cancers can be associated, most notably melanoma, as I've shown you, but also other malignancies, such as lymphoid pathway malignancies. So very fortunately for us, POP1 is an essential gene in HAP1 cells. And so this is a graph that shows the kinetics of cell growth of cells in which we've disrupted POP1. We've got two genes here that are non-essential genes, and also BRCA1, which is, of course, an essential gene. So because of this, this gives us an opportunity to use the Starita-Finley-based saturation genome editing approach. So what we did was to generate libraries of variants. This is an alanine scan, a stop codon scan, all variants that are found within ClinVar, within COSMIC, and all possible alterations within the gene. And so because we were generating libraries in this way, we used the HAP1 Lig4 cell line, and I'm going to describe this in more detail. And because this is an essential gene, we can use cell fitness as a biological readout in terms of examining the effect of those variants. So at Sanger, we've been trying to scale these approaches of saturation genome editing. And there are two things that we've developed that I think might be of general use to the community in terms of performing these experiments. The first of these is a software tool we call Valiant. And so Valiant is a tool that allows you to design all of those repair libraries that you can then use for saturation genome editing. The second is a cell line, and this is a HAP1 Lig4 mutant cell line. It's a cell line that retains a high level of haploidy. And in addition to this, it has a clonal insertion of Cas9 that is expressed at high levels. And furthermore, we've cloned this cell line, so it has a very high rate of homologous recombination. And in fact, the rate of homologous recombination is in the order of 50%. So this dramatically improves our ability to perform saturation genome editing at scale. So in addition to these two advances at Sanger, we've been trying to scale each of the different steps associated with saturation genome editing. And there are various posters that are available or on display at the meeting that describe these efforts. So the other thing that we've realized as we perform these experiments is that there's more information that is found within this variant data than the binary, is it disruptive or not disruptive? So to illustrate this, what I've got here is a time course where we've compared day 4 to day 7 and all these different time points down here to day 4 versus day 21. And when you look at this picture, you can see that there is different variant behaviors. For example, there are these variants here which deplete very quickly, and they deplete to a large degree. And they tend to be stop codons and also frameshift variants. And then there are these variants here which tend to be missense variants, and they deplete much more slowly. So it's not just 0 or 1, there's a gradient of variant effect that we can observe in these saturation experiments. So we've actually performed saturation genome editing across the entire gene, and this is the data shown here. Again, in keeping with what I showed you previously, the frameshifts and the stops are shown in yellow and red, and the missense variants are shown here in green. And the data is represented as a functional score, which is essentially a growth rate over the time period. So because we had this sort of data, what we can do is to use Gaussian mixture modeling to break the data down into different groups. And in this way, what we can do is identify a group of variants that we can call strongly depleted in dark blue, weakly depleted, a very small number of variants that we can see here are enriched, and a group of variants that appear to be unchanged. And this is what this translates to in terms of the different growth rates of these different groups. So then what we can start to do is to compare these different classes of variants to different sort of breakdowns of those variants. For example, these are the strongly depleted variants, which as you can see here are largely made up of those stops and frameshift, whereas the weakly depleted variants, as I've said already, tend to be those that are missense in nature. We can also expand this, for example, looking at the effect of those different classes of variants on different features, for example, on evolution. So you can see here that the strongly depleted variants have higher Eve scores in keeping with the fact that these variants tend to be more conserved. And as you can see here, the enriched variants tend to have lower Eve scores, which might suggest that there's some other biology that's operative. And we can do the same sorts of analyses comparing to, for example, data in Nomad or looking at splice effects. And you can see the distribution of splice effects for the different types of variants shown here. So just coming back to my colleague David, who I mentioned to you previously, who carries that I78T variant and has that family history of melanoma and CLL and CTCL as well. So this is the exon in which that specific variant falls. You can see specific variants that we know to be pathogenic in function that I've indicated here, and these are the associated malignancies here. And this is David's variant here. So it behaves like one of these strongly depleted variants, and therefore we would predict that this was a disruptive variant and therefore associated with his condition. Not only can we look at a variant like the one that David carries, but we can expand this across further pedigrees. These are examples of two pedigrees. This one is a Dutch pedigree where you can see there's co-segregation of malignancies together with this specific variant, which is disruptive of function in our saturation genome editing experiments. This specific variant here, we may well have suspected could be disruptive, but this variant in this family here is a synonymous alteration, and we certainly wouldn't have predicted that this was disruptive of function. So the fact that we're generating all of that different nucleotide level variation across the entire gene gives us an opportunity to explore variants like this. And in keeping with this family history here, this variant we can also find is disruptive. So the other thing that we can do with these sorts of data is to compare the information that we obtain from our saturation genome editing to large-scale population-level phenotyping data sets. And one good example of this is UK Biobank, where there are 500,000 individuals who've been very, very extensively phenotyped across a range of different phenotypes. So the phenotype, the cancer phenotype that is most common within this population is actually blood cancer. And so if we compare the risk of blood cancer to each of the different types of SGE-associated variants, those people who carry disruptive variants within POT1 have an increased risk. But what's possibly more informative is what's shown here on the right-hand side, where the strongly depleted variants are associated with around a 6-fold increase in risk of developing a blood cancer relative to the general population. So this kind of information can be used for example, risk prediction modelling. Another phenotype that is also of relevance and found within UK Biobank is telomere length. And this has been calculated for all of the individuals that are within this data set. And so what we can find is that the variants that are disruptive by SGE are associated with longer telomeres. And this is in keeping with the overall biology of this disease that I've mentioned to you previously. So what I've tried to illustrate to you is that these kind of saturation genome editing experiments can be hypothesis generating. And it's really as a result of the work that we've done on POT1 that we've come to believe that there are sort of two different phenotypic readouts in the context of this gene. One phenotypic readout is a high risk of developing cancer, and that's associated principally with stop codons and also frameshift variants. And then there's a more intermediate phenotype, and this is associated with missense variants. Those individuals also have longer telomeres, but they're less predisposed to developing the disease. So in addition to working on POT1, we've also been exploring other genes. For example, we've recently published work on both BAP1 and also RAD51C, and these are the individuals shown here, many of whom are in the audience, who have led the work that I've described in these papers, or that we've described in these papers. So what we plan to do over the next few years is to really take on the challenge of exploring all of the cancer predisposition genes that are routinely tested in clinical practice. There are roughly around 114 of these different genes. And if you go to ClinVar, there are around 50,000 variants of uncertain significance. Importantly, those 50,000 variants represent not 50,000 individuals, but in many cases, tens of thousands of individuals. And if you consider that ClinVar is composed largely of people of European descent, and you extend across other populations, it's likely that there are many millions of individuals who would benefit from a genetic diagnosis or an enhanced genetic diagnosis, which would mean that they could potentially receive screening. So this is really our agenda for the next 5 to 10 years. So in saying that, we're very mindful that most of the work we've done so far has really revolved around exploring life and death. But very clearly, cellular phenotypes are more than just life and death. So because of this, what we've been doing is trying to develop new technology that allows us to examine variants in different ways. And possibly the richest phenotype that we can come up with is single-cell gene expression profiles. And so because of this, we've developed a new method that we call Saturation-seq. So the gene that I'm going to describe where we've used this method is a gene called NRF2. NRF2 is associated with liver cancer, esophageal cancer, and also cancers of the lung. And it tends to be mutated in two specific regions of the gene, which are found at the N-terminus, which are called the DLG and the ETGE region, as indicated on the slide. Just one slide of biochemistry. What happens within a cell under normal situations is that NRF2, which is shown here, will bind to this protein called KEAP1. And when this occurs, NRF2 will be degraded, which is what's happening here. If you disrupt KEAP1, or alternatively you mutate NRF2, this can result in an increase in the levels of this NRF2 protein, which can then translocate to the nucleus, and it can activate gene expression and allow the cells to survive. So the question that we invariably get asked whenever we perform one of these sorts of experiments, particularly when we do it in HAP1 cells, is the biology of HAP1 relevant to this phenotype? So this particular experiment here examines that pathway, the NRF2 pathway, in these HAP1 cells. So what we've done here is to disrupt KEAP1, as you can see. This results in an increase in the levels of NRF2 in keeping with what's shown on the left-hand side, an increase in the level of a downstream target gene, which is called NQO1, and ultimately this is also associated with an increase in the growth of these HAP1 cells when we do plating-based assays. So this pathway is operative within these HAP1 cells, and therefore we can screen for the effects of variants within NRF2 within this cell line. Clinically, we know that patients who carry mutations within NRF2, that they are more resistant to a chemotherapeutic drug called cisplatin. And so if we introduce specific mutations into NRF2, as you can see here, this is associated with a shift in the resistance to cisplatin, as you can see here. So this is in keeping with this pathway being operative within the HAP1 cells. And similarly, we can see these gene expression profiles, which is in keeping with this pathway functioning in this way. So how does Saturation-seq work? So we take our HAP1, LIG4, Cas9 cell line that I mentioned to you previously, highly clonal, high level of Cas9 activity. We introduce into those cells this vector shown here, which expresses a barcode. And so we can then sort those cells, so we have a population of cells that are highly haploid, where each cell in the population contains a different barcode. What we can then do with those cells is and then in this context, what we're interested in doing is examining those two regions, the DLG and the ETG region. And so there are around 630 variants within that region that we want to examine. So what we can actually do in that way is then perform a standard saturation genome editing based experiment where we design one of these repair libraries. We introduce these into the cells. We can then take each of the single cells that are produced from that experiment, and half of the DNA and RNA can be separated so that the locus, the NRF2 locus, can be genotyped on the Tapestry platform, and we can generate single-cell sequencing data on the 10x platform. So that gives us an opportunity to correlate or to compare the specific variant that we've introduced into the NRF2 locus together with the 10x single-cell expression profile. So when we do this, we end up with the heat maps that you can see shown here, or the U maps that you can see. And there are very clearly two populations, this one here in blue, and this other one shown here in red, for both of these two different regions that we've saturated. And if we do differential expression between these two different groups, you can see the pathways that are altered, including the NRF2 signaling pathway and other downstream effectors of this pathway, suggesting that these variants that you can see in red are variants that are altering this pathway. So importantly, what we can then do is we can drill down right on specific variants. For example, at each of these different amino acid positions across the gene, we can look at the individual variants and their effect on NRF2 function. And this is expressed as a disruption score, which is essentially a gene expression signature that is associated with alteration of NRF2 pathway and signaling. So this gives us very rich and granular data to explore the effect of introducing variation into the NRF2 gene. And this is just an example of these sorts of data where at each of the individual positions across that region of NRF2, we can look at genes that are differentially expressed. And Magda Strauss is going to talk later today a little bit more in detail about how we get to these sorts of differential expression analyses and identify these genes and pathways. So NRF2 is also not only involved in cancer, but involved in a neurodevelopmental condition. And importantly, if we look at specific variants that are found and that are disease-associated, such as these two here, these are also associated with a high disruption score. So we think that in addition to exploring variation in the context of cancer, we can use these sorts of saturation genome editing data of NRF2 to examine phenotypes that are associated with this neurodevelopmental condition. So this is really a new method. We're trying to work out how we can scale it. We think that it has a particular opportunity or a sweet spot that complements other methods, such as, for example, prime editing, where you can also edit the genome and get a single-cell expression profiling. And possibly its particular strength is really for examining complex alleles, or where you want to introduce multiple events into the same locus of a gene, which is problematic using other approaches or methods. So I wanted to finish there and thank the people who've done the work. The majority of the studies, the saturation genome editing studies in my group, are led by Andrew. The POT1 work was led by Sophia. Rebe did the RAD51C work. Our human genetics work is performed together with Yaji, and Magda, who will talk later, was heavily involved in the NRF2 saturation genome editing studies. Thank you.